Adjust the brightness. Haji, so good morning everyone. How are all of you? Am I audible, visible? Pin your response. Good afternoon, good afternoon. It's all right, everything done? Okay, ji. So probably in last class we have discussed about some of the composition of the plasma membrane, and now we have to study about which one? Structure of plasma membrane, right? So we are starting with the structure of the plasma membrane. A structure of plasma membrane. Okay. So if we talk about the structure of plasma membrane, there are so many different models given by different scientists. But out of that all given model, the best accepted model is given by Singer and Nicholson. In 1972. Right. And what model he have given? The fluid mosaic model. Fluid mosaic model. This fluid mosaic model given by Singer and Nicholson. Now question arises, what is the fluid mosaic model? Fluid here represent the continuous part of plasma membrane. Or you can say liquid part of the plasma membrane. And mosaic here is what? Discontinuous part of plasma membrane. Or solid part of plasma membrane. Got the idea? So one is liquid part, another is solid part. So plasma membrane is made up of two type of component. One liquid component and another solid component. But who is making the continuous structure of the plasma membrane? Liquid component. And who is forming the discontinuous structure of the plasma membrane? It is solid component. Now, I am just giving you two examples to understood better about which one? Fluid mosaic. Try to understand. One example is gulab jamun in sugar syrup. One example is what basically? Gulab jamun in sugar syrup. You assume sugar syrup where gulab jamun are placed. You have seen in the sweetmeat shop. So big container is there, and in that container so many gulab jamun they are placing. So who is liquid part? Sugar syrup. Who is continuous part? Sugar syrup. And this sugar syrup is equal to lipid. This sugar syrup is equal to lipid. Got the idea or not? You think like assume sugar syrup of the gulab jamun is equivalent to which one? Lipid part of the plasma membrane. And gulab jamun, they are embedded here, here. Some are deeply embedded, some are superficially embedded. Got the idea? R protein. So how they are embedded, how the plasma membrane look like, you got the idea? In a liquid part, some solid part is embedded in a mosaic-like fashion here and there. If I give you a rough sketch, you think like this is a continuous liquid part that is lipid bilayer and some protein is embedded like this. It is gulab jamun, another gulab jamun, another gulab jamun, another gulab jamun. Are you getting the idea or not? Another gulab jamun. It is so, and it is layer of sugar syrup. It is sugar syrup. Are you getting the idea? These are gulab jamun. So these are the liquid component, these are the solid component. So these are which protein? Extrinsic protein. Extrinsic protein. It is intrinsic, but superficial intrinsic. It is intrinsic, but totally buried. I told about this protein, na? Totally buried. Or it is which protein? Transmembrane protein. So these continuous white line is made up of lipid bilayer, and these are in mosaic-like fashion. Who is present? Solid protein is present. So such kind of a structure, getting the things or not? Superficially means not totally deepen. It is what basically? A very outer layer, na? You just see just an outer extrinsic protein. And here intrinsic superficially, better to write partially. Better to write here which word? Partially, not totally. Partial and total is the correct word. So look, here it is just embedded. You can easily extract this. And who can easily extract is called extrinsic or peripheral protein. Peripheral protein. Got the idea? It is deeply buried, but not totally buried. So it is difficult to isolate it. So hence it is intrinsic, but it is partial. It is intrinsic, but totally buried. So it is called what basically? Intrinsic and totally buried, and it is called transmembrane protein. I told na in the last class that there are two type of protein, is there extrinsic protein and intrinsic protein. So here this is extrinsic, this is extrinsic, and these all three are intrinsic. It is partially buried and it is totally buried. Partially buried in the membrane. Got the idea? Picture came in the mind. Is it all right? So another example to understand the plasma membrane structure is iceberg in an ocean. Iceberg, solid, protein, ocean, liquid, lipid bilayer. Understanding the idea? Picture came in the mind or not? Is it all right? It is done. Now I am moving ahead. So now I am drawing the structure of the plasma membrane. You just try to understand. This is lipid bilayer. Head is both outer side, tail is in the center of the membrane. Got the idea? It is like this structure. And you will find, assume here, you just note, pin which kind of protein I am making here. It is extrinsic or intrinsic, superficial, partially or totally buried. Got the idea or not? And then I am making which one? So who is embedded in between? The liquid component, solid component, and that is the protein component. Understanding the idea? I am making one more protein here. You just try to understand. I am making one more protein here. Got the idea? Now, this white color part only is what basically? Lipid bilayer. Two lipid is there. That is the reason I am telling it is what? Bilayer. I am writing it is outside of cell. It is plasma membrane. Thickness is 75 angstrom. It is inside of cell. Inside. Inside is what? Cytoplasm. So this is the plasma membrane. And meanwhile, another type of protein is also there. Some protein is like this, like this. Some protein is like this. Some protein is like this. Got the idea or not? So protein present in a which one? Mosaic-like pattern. No any continuous pattern of the protein is there. So who is embedded in a mosaic-like fashion? Protein. This is extrinsic protein, intrinsic partially buried, transmembrane protein, intrinsic totally buried, intrinsic partially buried, extrinsic protein. Are you getting the idea? Understanding came about the things or not? Tell me, first you tell me yes or no. Picture came in the mind. So how the plasma membrane structure is there? Lipid bilayer forming the continuous structure of plasma membrane, and protein is present in a which one? Mosaic like fashion. And I told all different type of protein. Who can easily remove is extrinsic or peripheral protein. Who can't easily remove is called intrinsic or integral protein. Intrinsic protein is two type, partially buried and totally buried in the membrane. This one is also called transmembrane protein. Got the idea? Now give your attention. So plasma membrane have a quasi-fluid structure. So I am writing here the quasi-fluid a structure of plasma membrane is due to phospholipid. Who is behind the quasi-fluid nature of plasma membrane? Phospholipid is behind the quasi-fluid. Quasi-fluid means what? Plasma membrane is not a solid structure. It is a fluid-like structure. It is like honey layer. Layer made up of honey type of thing, viscous. Got the idea? It is not too much liquid, but not too solid. It is somehow quasi-fluid, honey-like. Getting the idea or not? Understanding the idea or not? So, who is behind quasi-fluid nature of the plasma membrane? Examiner will ask. Phospholipid is behind the quasi-fluid nature of the plasma membrane. And what is the advantage of quasi-fluid nature? So, quasi-fluid nature of plasma membrane quasi-fluid nature of plasma membrane help in lateral movement. One work. Another work I am writing where quasi-fluid nature help. It help in cell growth. Another area it help in cell division. Another area it help in formation of intercellular junction. It help in endocytosis and it help in cell secretion. So these all are the work performed by a cell become possible due to which nature of the plasma membrane? Quasi-fluid nature of the plasma membrane. Understanding the idea or not? These all work is due to quasi-fluid nature of plasma membrane. Now let me explain. Usually you people mug up, don't understand the meaning of lateral movement. Let me tell you. Suppose that here this board is a brick wall. It is a brick wall and I am taking a nail and hammer and hitting in the brick wall. What happened? This nail will pierce in the brick wall, move inside the brick wall. After few minutes I realize I put the nail at the wrong place. Here I put, but I actually want the nail here on the ball. Is it possible for me to drag the nail like this? It is not on the good place. I am dragging on the wall and nail shifted from one area to another area. Tell me, is it possible or not? Is it possible or not? Not possible. Because this is called lateral movement. You got the meaning of lateral movement? You got the meaning of lateral movement? Who not allow the lateral movement? Solid do not allow the lateral movement. Because intermolecular space is very less. It not give the passage to move the things. Getting the things or not? Understood? So lateral movement not allowed by which one? Solid. But look, liquid. Plasma membrane allow lateral movement, na? And plasma membrane is made up of honey type of material. Which type of material? Honey type of material. So what I am taking? I am taking a container where honey is there. Now I am just dipping it 50%, half of the pen into which one? Honey. And then I am doing like this. Is it possible to do like this? Drag laterally. Possible, is it? Possible or not? Tell me. In honey, it is possible. So it is called which movement? Lateral movement. Who allow the lateral movement? Who allow the lateral movement? Liquid allow the lateral movement. Solid not allow the lateral movement. Now you can say, sir, why you are talking that much about lateral movement? I am telling you the reason. You have studied about facilitated diffusion, na? Facilitated diffusion, na? So in facilitated diffusion you have studied, na? NCERT wrote also in transport कि भाई protein rotate 180 degree in the membrane. Now this is the protein, you try to understand. This is a protein, this is rotating by 180 degree during facilitated diffusion. Upper part came lower part. So how it is possible? This is going to this side, then going to this side. Is it possible without lateral movement? Is it possible without this rotation is possible by 180 degree without lateral movement? If they are not allowing lateral movement, it can't bend like this, na, and came this side, na. So it is possible due to which one? Lateral movement. Got the idea? Is it clear in the mind? Now, I am telling you one thing. When I was a student like you, my teachers weren't teaching me amoeba. So when they teaching amoeba, they are telling the function of contractile vacuole. Ki look, in amoeba, contractile vacuole is there. And these contractile vacuole perform osmoregulation. So when excess of water entering in the amoeba, they store inside the contractile vacuole. Slowly contractile vacuole move near the plasma membrane. They hit the plasma membrane, excess water came out. And in such a way, contractile vacuole perform the osmoregulation. Got the idea? So as a student, that time in my mind a question arises, कि भाई, if a contractile vacuole hit the plasma membrane sir is telling break and water will come out. If plasma membrane break and water will come out, water will also come in, na? If it is breaking anything, water is going out, water may come in. So what is the benefit of this? That time, I not dare to ask that question to my teachers. Got the idea? But later on when we studied more, we find it is possible due to quasi-fluid nature of plasma membrane. Plasma membrane can easily break. It is fluid, na, liquid, na. I am giving you a stick and telling beat the water. When a stick came in contact with the water, who will separate from this area? Water. As you remove the stick, who will join together? Water. Who will join together? Water join together. Understanding the idea or not? Picture came in mind or not? One of the student writing video not coming. Not clear. It may be your network issues. Everywhere not clear? Voice breaking, video not coming, it's clear. I think it's problem on the app. So problem on app, so you can shift on YouTube. Who have problem? I think right now for few more days, the classes is still on YouTube. So you shift on YouTube. Right? I think YouTube people writing it is clear. App it is saying it is not clear. Okay, so you shift on YouTube. Clear? Understood? So got the idea what I want to convey you? Plasma membrane can easily break and easily join. As it break, as it join. As something is there in between, they will break. As it will remove, it will join. Got the idea? Is it possible in the Na? So you got the idea about the plasma membrane. Which one? Quasi-fluid nature and advantage of this quasi-fluid nature. You got the idea, picture came or not? Now I am going to tell you transport across the membrane. Transport across the plasma membrane. So you know about these things? Cell growth, liquid na cell growth occur, cell division occur, formation of intercellular junction, Anurag sir will teach you. Gap junction, tight junction in anatomy, tissue. Able to understand. Endocytosis, when a cell takes something in from outside, it is called endocytosis. Endocytosis. These endocytosis is of two types. Phagocytosis and pinocytosis. Phagocytosis when cell takes solid food, solid material inside, and pinocytosis when cell takes liquid material inside. Phagocytosis is also called cell eating, and pinocytosis is also called cell drinking. These all is possible due to quasi-fluid nature of plasma membrane. Cell secretion, these all are possible due to quasi-fluid nature of plasma membrane. Now what we are going to talk? Now we are going to talk about transport across the plasma membrane. Transport across the plasma membrane. So if you talk about plasma membrane, through plasma membrane, two type of transport occur. One is passive transport. One is which transport? Passive transport. Another is which transport? Active transport. Active transport. So passive transport occur toward the gradient. Toward the gradient means from higher to lower concentration. And active transport occur against the gradient. Against the gradient. That is from lower to higher concentration. Anything when move from lower to higher concentration, it is called active transport. Anything when move from high to low concentration, it is called diffusion. Now, don't require energy. ATP you can write. Here, active transport require ATP. Understanding the idea or not? Now, if I told about passive transport, two type of passive transport occur through membrane. One is simple diffusion, and another is facilitated diffusion. Another is facilitated diffusion, which occur through plasma membrane. So try to understand simple diffusion. If you talk about this simple diffusion, who going for simple diffusion? It occur through lipid bilayer of plasma membrane. Occur through which one? Lipid bilayer of plasma membrane. And this simple diffusion, who go for simple diffusion? Neutral particle. Example, neutral substances. Hydrophobic substances. Another you can say about non-polar substances. Transport across the membrane with the help of which one? Simple diffusion. Now facilitated diffusion These facilitated diffusion occur with help of carrier protein. Carrier protein. An example who go with facilitated diffusion, I am writing, example is charge particle. Charge particle not allowed by lipid bilayer. You can say polar substances not allowed by lipid bilayer to cross. So they take the help of carrier protein or transmembrane protein. Another things you can say about hydrophilic substances. Hydrophilic substances. They are transporting across the membrane by which diffusion? Facilitated diffusion from their higher concentration to lower concentration. These are the things by simple diffusion. These are the things with the facilitated diffusion. Protein, membrane protein involved here, membrane protein not involved here, and active transport I am writing now. Now I am writing which one? Active transport occur by pump protein. This active transport occur by pump protein. Pump protein. And it better know I wrote already against of gradient. It is occur against of gradient. An example is sodium potassium pump. Sodium potassium pump is an example of active transport. Got the idea? So you got simple diffusion, facilitated diffusion. Now one note I am writing, diffusion of water is called osmosis. Diffusion of water is called osmosis. Diffusion of water is called osmosis. So who is over? Plasma membrane is over. Understand the idea? Picture came in mind. Should I ask a small question, 2, 3? Can you answer it? And then we move toward which one? Endomembrane system. Look here. Now I am asking a question based on the diagram. Here is the diagram. Let me ask a question. Tell me, when anything moving from high to low concentration like this, it is which kind of transport? High to low concentration. High to low concentration. Passive. Simple diffusion or facilitated diffusion? Simple or facilitated? Simple. When something coming at the protein binding with it from high to low concentration and transporting then high to low, it is facilitated diffusion. And something from low to high, this side more, this side less, binding here and passing here, it is called active transport. And one more thing I am telling you, do you know where carbohydrate present in plasma membrane? Carbohydrate is present here. It is oligosaccharide. It is oligosaccharide present on outer surface. It is oligosaccharide. So when the carbohydrate present with protein, it is forming which one? Glycoprotein. And when it is attached with lipid, it is called glycolipid. So glycoprotein and glycolipid in plasma membrane act as receptor. These glycoprotein and glycolipid act as receptor. A receptor and always present toward outer side. So who is over? Plasma membrane is over. Got the idea? Can we move ahead? Done. Now we are moving toward endomembrane system. Endomembrane system. So when you look at a eukaryotic cell, in a eukaryotic cell, so many cell organelles are there. In eukaryotic cell, what is there? So many cell organelles are there. And each and every cell organelles are distinct. They have a specific structure and function. So what I am writing, each and every cell organelle present in eukaryotic cell have a distinct means a specific, separate, a structure and function. Distinct structure and function. Got the idea? So each and every cell organelle present in eukaryotic cell have distinct structure and function, but some of the cell organelle have coordinated function. Means their function is interconnected. That is interconnected function. Coordinated function. And these cell organelles are called membrane system. These cell organelle are called which system? Endomembrane system. So it include first cell organelle ER, then Golgi complex, next is lysosome, and next is vacuole. So these are the cell organelle all together called endomembrane system because these cell organelle have coordinated function. Cell organelle like mitochondria, plastid, or you can say chloroplast, not a problem, chloroplast and peroxisome not have coordinated function. Hence it is not a component of endomembranous system. Endomembrane system. Are you getting the idea or not? So, who are endomembrane system? That cell organelle who have a coordinated function. The cell organelle who don't have a coordinated functions is not a component of endomembranous system. So, in eukaryotic cell, these four cell organelle have coordinated function and they are called which one? Endomembrane system. Mitochondria, peroxisome, peroxisome, chloroplast not have a coordinated function and hence it is not a component of endomembrane system. You got the idea? So I think this is over about endomembrane system. How the coordinated function is there, I will teach later after teaching all the component. Now I am starting with endoplasmic reticulum. I am starting with which one? Endoplasmic reticulum. In short, we called it ER. So what NCERT wrote about ER? When you read your textbook, it is written like कि when you study the eukaryotic cell with the help of electron microscope, when you study the eukaryotic cell with the help of which one? Electron microscope, you will observe कि in the eukaryotic cell there is a network or reticulum of tiny tubular structure, and these network of tiny tubular structure present in the cell, scattered in the cell, is called endoplasmic reticulum. You got the idea? So I am writing this line in short. It is the network of tiny tubular structure present in, or better to say scattered in, cytoplasm. Scattered in cytoplasm. And this is called endoplasmic reticulum. If you want to see the structure of it, just give your attention toward this diagram. So this is the nucleus. This is the nucleus, and this is plasma membrane, and this structure is called nucleus. So now what you will find, from this nucleus itself, who is extended, there is a membranous tubular structure is arising, and these membranous tubular structure connect one side with nuclear membrane, another side with which one? Plasma membrane. And these structure which form here is called endoplasmic reticulum. Are you getting the idea or not? So if you look very carefully to the endoplasmic reticulum, you will find that the part of ER near the nucleus have rough or granular surface due to presence of ribosome, and it is called rough ER, rough endoplasmic reticulum, and part of here poor toward plasma membrane, that ribosome is not there, and that is called SER, smooth endoplasmic reticulum. So are you able to understand that these are a network or reticulum of tiny tubular structure scattered in the cytoplasm? See now how? It is one side attached with nuclear membrane, another side attached with plasma membrane. Got the idea? So toward nucleus side, ER have granular surface or a rough surface, because this side over ER ribosome attached, and it is called RER, but toward plasma membrane ribosome don't present, and that is called, it is SER, smooth endoplasmic reticulum. So now I am moving ahead about the endoplasmic reticulum. So ER divide cytoplasm into two compartments. Who divide into two compartments? ER divide the cytoplasm into two compartments, and the name of this compartment is, one is luminal and another is extraluminal. So what is luminal? Cytoplasm present inside the lumen of ER. Lumen of ER is called luminal. And what is extraluminal? Cytoplasm present outside of lumen of ER. is called extraluminal. I am just making a marking so you will understand it better. So you just tell me, I am making a cross here. It is luminal or extraluminal? And here it is what basically? So this cross is luminal inside the lumen, and this cross is what? Outside of the lumen. Understanding the idea or not? Is it there? Clear in the mind? So who is dividing the cytoplasm into two components? Getting the things? ER dividing the cytoplasm into two component. Understanding the idea? What is the name? Luminal and extraluminal compartment. Got the idea? Can I move ahead about the ER? Just have a look here. Now I am moving ahead on the ER. So now I am just talking about component of ER. This line not in NCERT, but I am giving a brief account. Component of ER. So endoplasmic reticulum have three different component. Three different component. And out of three different component is one component is cisternae. Another component is vesicle. And the third component is tubule. So cisternae is a flattened stack like a structure. Flattened, unbranched, flattened structure. It look like this. It is cisternae. It stack over each other. Vesicle is what basically round vesicular structure. And tubules is branched and tubular structure. It is branched, tubular and irregular structure. So it is tubule. So it is three component of which one? Endoplasmic reticulum. In these all three component also present in Golgi body. But in Golgi body they are present as a complex, and in endoplasmic reticulum all these three component of endoplasmic reticulum is remain separate. Understanding the idea? So these are the three component of endoplasmic reticulum is there. Cisternae, vesicle and tubules. Now I am going to talk about the type of ER. So if you talk about the endoplasmic reticulum, it is of how many type? Two type. One is rough endoplasmic reticulum and another is smooth endoplasmic reticulum. Now I am going to tell you some differences between rough endoplasmic reticulum and smooth endoplasmic reticulum. Now RER is what basically? It is granular. Here it is agranular. Here ribosome present. This one ribosome absent. It is present toward nucleus. It is present toward plasma membrane. Short I am writing. Now cisternae is more. Is more in which one? RER. In SER, tubule is more. RER abundantly present in cell associated with protein synthesis. Associated with protein synthesis. Who is abundantly present in cell? RER abundantly present in cell associated with protein synthesis. ACR, it perform, got the idea? It perform lipid and steroidal hormone synthesis. It performs lipid and steroidal hormone synthesis. Who do the lipid and steroidal hormone synthesis? ACR. And now don't confuse here. If anybody going to ask a question, कि protein synthesis cell organelle associated with cell organelle perform protein synthesis is RER, ACR, ribosome and lysosome. What answer you will give? Cell organelle perform protein synthesis is? Answer is ribosome. RER not do protein synthesis. This is the RER. Here ribosome is present. This ribosome doing the protein synthesis, which is present on which one? Endoplasmic reticulum. Got the idea? But who do the lipid synthesis? ACR do the lipid synthesis and steroidal hormone synthesis. RER not do protein synthesis. Ribosome present on rough endoplasmic reticulum doing the protein. Getting the idea or not? Ribosome doing the protein synthesis, not the ER. Because ribosome present on ER, so the site of protein synthesis is ribosome. But it is performed by whom? Site of protein synthesis is what basically? Ribosome present over ER. Got the idea? Clear in the mind? So don't confuse here. So who is over? Difference between RER and SER is over. Now I am going to tell you some additional function of ER, which is not in NCERT, but it is important to know. Intracellular transport. Formation of phospholipid and cholesterol performed by which one? Smooth endoplasmic reticle. Another job done by ER is formation of phospholipid and cholesterol performed by which one? Smooth endoplasmic reticulum. Formation of lipoprotein and glycogen also done by ER. Another function I am writing, glycosylation. Glycosylation also performed by ER. Actually, glycosylation is the main function of Golgi body, but it is not written in which one? ER in NCERT. And glycosylation word is also not written in which one? No. Formation of phospholipid occur in ER, a smooth endoplasmic reticulum. Formation of phospholipid occur in which one? Smooth endoplasmic reticulum. Lipoprotein and glycogen formation occur by which one? ER. Glycosylation also performed by ER. Glycosylation, during glycosylation what happened? Formation of glycoprotein and glycolipid. And what I am telling you, understand, try to understand carefully. I wrote what? Also, formation of glycoprotein and glycolipid mainly done by Golgi. NCERT wrote in Golgi body. Got the idea? What I want to say, try to understand. 4 years back, probably 2017, a question came in NEET. Which cell organelle is associated with glycosylation? And it is not written in NCERT. Glycosylation word not written anywhere in NCERT. But question came in NEET. In which year? 2018. What was the question? 17. Glycosylation occur in which cell organelle? And who was not in option? Golgi body. Who was in option? ER. If I not told you this line, are you able to do that answer? It become difficult, na? Getting the idea? That's the reason I told this line. If coming option is both which one? ER and Golgi body, you opt for Golgi body. But if Golgi body is not there, you opt for which one? ER. Mainly done by Golgi body. But who also have a role? ER in glycosylation process. Are you getting the idea? Picture came in the mind. Now another function of ER I am writing. Another function of ER I am writing. That is what? That is formation of Golgi body. Formation of Golgi body. This is also the function of which one? ER. So these are the some of the additional function of ER I told. Got the idea? And today up to here only. Okay, ji, right? So in your batch it required to take one extra class. Reason? You just recall 2 times our class is not occur in your batch. One due to mic problem and one network was not there. If you are able to recall. Got the idea? So this week we are giving entire cell biology. So for your batch, I will take any day extra class. I will inform you in advance. Got the idea? Okay, ji, bye-bye. Have a good day to all of you. So you understand the ER? So in next class, probably we will finish Golgi body, lysosome, vacuole, as well as mitochondria. That much we finish in that, probably the extra class if it is coming. Okay, ji, bye-bye. Bye, 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 bye.